It's my pleasure now to introduce today's speaker. I think, with the exception of very long-lived married couples in this audience, I have known Jeff Bada longer than anyone else in this room has known anybody else. Jeff and I went to high school together. And you can see the effect of us going to high school together is that, here's Jeff. When I first came back to UCSD in the early 70s after being at the Smithsonian for a number of years, I was told about my colleague and my friend from high school who had come to UCSD and gotten his PhD in chemistry in 1968, and then had gone to Harvard for a couple of years and was hired back to the faculty. And someone said, well, he just had his picture in a little article about him in Time magazine, and that seemed like a good reason to make him an associate professor really fast, and so that was happened. And his career has been meteoric. He's gotten a lot of notoriety for his work in the ability to use amino acids to age animals and to determine the age of whales as being over 100 years, and using specialized techniques that he has invented and established as ways of using short-term chronometers in the form of amino acid, the natural rotatory capabilities of these organic molecules over time. That, however, is not the subject for today, because Jeff has gone beyond just simply looking at questions about amino acids on this planet, but is actually going in the direction of looking at a subject called exobiology. And exobiology is the study and the looking for life in places other than on the Earth. And you can read about that in this book, which was written and published last year, The Spark of Life, Spark of Life, Darwin and the Primeval Soup, written by Christopher Wills and Jeff Bada. Jeff has had a very distinguished career at Scripps since he returned here in 1971. He is now the director of the NASA Center on Exobiology. And the subject for today is Bring 'em Back Alive, in which he's going to tell you about the search for life on Mars. So, Jeff, it's a pleasure to have you here.
Well, thank you very much, and it's indeed a pleasure to tell you a story about where we stand with respect to looking for life elsewhere. One of the fundamental questions, I think, that preoccupies humankind is the question of, are we alone? And this has been an area that's become extremely interesting over the last several years, because we found that there are other solar systems out there other than just our solar system, and there are extrasolar planets. And so the question of life elsewhere has taken on new importance. In our solar system, of course, we have these inner planets, the Earth being one, and then we have these gas giant planets on the outer parts of the solar system. With respect to life in our solar system, the only place we know that life exists is in this little blue dot right here called Earth. But there's been a lot of speculation that there could be life elsewhere. Early in the history of the solar system, Venus, which is basically our sister planet, may have been habitable. Today, however, Venus is a scorchingly hot world with surface temperatures on the order of 600 degrees centigrade. And so the potential for any kind of living entity as we know it existing on Venus is essentially zero. The next planet, however, Mars, is quite interesting, because there's a lot of speculation that Mars may have had a much different history than we see when we look at the planet today, and it's possible that life may have began on that planet as well. And the only other place that's considered a potential place for life is on one of the moons of Jupiter, a moon called Europa. I'll show you a picture of this later. But Europa is an incredibly interesting place because it's about the size of our moon, but it's covered with liquid water ice. And calculations suggest that underneath that ice is an ocean 100 kilometers deep. And if you go and you calculate the volume of that ocean, it's equal in size to our ocean. And so another possible place where life might exist is out here in this remote area around Jupiter. Here's Earth. You look at Earth from space. This happens to be a picture taken by the Apollo astronauts. The thing you see right away, it's a blue planet. It's covered with liquid water. And one of the essential things that is required for life as we know it is water. Life cannot exist without water. And so one of the goals as we look at other worlds is to try and see where we see the presence of liquid water. Now, if you look at Mars today, Mars is a barren desert world. There's no blue color to it, it's red, and that red is rust. And so the possibility that life may exist here, originally it was thought to be zero. But we now know that early in the history of Mars, it probably looked a lot different. And this is a reconstruction of what Mars may have looked like some 3.5 to 4 billion years ago. And you can see there's not global oceans like there are on Earth, but there's still large bodies of standing water present on the planet. And the brave soul that drew this picture painted some green color in here. And of course, green color means photosynthetic organisms. And so this person was being very optimistic that not only was there life here, but it was photosynthetic based life. And that has interesting implications because on Earth, all of the oxygen that's present in our atmosphere is a product of photosynthesis. And so whenever you have the potential for photosynthesis, you have the potential for oxygenating the atmosphere of the planet. So one of our goals is to try and figure out whether there ever was life on Mars and whether life might still exist today in some remote area. Now, if we look at the scenario by which we think life began on Earth, the basic process by which life began on Earth is to start out with some sort of natural chemistry that produced the kind of molecules that are associated with living organisms, amino acids, the compounds that make up our genetic material, DNA and RNA. Those compounds could be made directly under simulated early Earth or early Mars conditions. The other thing we know is that certain kinds of meteorites and comets contain a wide assortment of organic molecules. Many of these are the kinds of organic molecules that play an essential role in biology. So the idea was that by either direct homegrown reactions or the infall of material from space, we ended up with a prebiotic soup, a soup of organic material that consisted of simple molecules, but those molecules were by and large the kind of molecules that are used in biology today. Those simple molecules underwent polymerization. In other words, they went from simple monomers to more complex polymers, and you started growing an inventory of more and more complex molecules. And out of this gamish of complex polymers, something appeared that satisfied the criteria for the first living entity, and I'll show you what that is in a second. And so this is the point of the origin of life. Now on Earth, this evolved first into the RNA world, a time when life on Earth consisted nothing more than self-replicating RNA molecules, and that then evolved into the protein DNA world, which characterizes all modern biochemistry. So we're protein DNA organisms, and that's the biochemistry that evolved from whatever this first living entity was. On Mars, we could have, again, the origin of life, but we have no real understanding of how that life may have evolved in a completely different environment. Now, what do we mean by life? A lot of you think of life as something that you can see or has a cell or whatever, but when we're talking about the origin of life, we're talking about nothing more than an autonomous, self-replicating system that evolves by natural selection. Now that's a fancy way of saying that you need replication. This molecule has to replicate itself, and that replication is imperfect. And the reason the imperfect part is important is that you'll have then errors that occasionally accumulate in the molecule. Some of those errors may be advantageous. It may give the molecule an added ability to, say, bind something or do something. And that molecule will then evolve and become the dominant one in the system. So with random errors, you also start the process of evolution. So the origin of life, of a simple self-replicating entity, is synonymous with the origin of evolution. So remember, when we're searching for life elsewhere, we cannot assume that we are going to have evolved entities like we have on Earth. It could be very simple molecular-based life, and we have to be able to assess whether that kind of life is present. Now, if we look at the search for life on Mars, it has a very long history. At the first part of the 20th century, Percival Lowell, who was a very noted astronomer, was observing Mars with his new fancy telescope at the Kitt Observatory in Arizona, and claimed to see canals on the surface of Mars. And Lowell argued that these canals were constructed by intelligent beings, and these canals were used to transport water from the polar regions of the planet, where the ice caps are, to the equatorial regions where there's no apparent source of water. And Lowell was so pervasive in these arguments that a large fraction of the scientists at the time believed not only was there life on Mars, but there was intelligent life. And this reached its climax in the summer of 1924, when a group of scientists went to the US government and asked that all radio broadcasts be stopped in the United States for a period of 48 hours, because Mars was as close to the Earth as it had been in 25 years, and it was as close as it was going to be for another 50 years. And so the idea was, with the close proximity of the planet, and if there were intelligent beings there, maybe we could hear their radio broadcasts. So all radio broadcasting in the United States was stopped for 48 hours, and these listening devices were placed to focus on Mars. And guess what? They heard a repetitive signal that made front page news on the New York Times that there were Martians trying to communicate with us. They brought in some of the best code breakers of the world to try and decipher this signal. And finally, one chap says, "I've heard that before. It's a transformer shorting out." And they finally found the transformer in upstate New York. So the Martians were right in our backyard, so to speak. In 1938, a very famous event took place. Orson Welles read H.G. Wells' book, The War of the Worlds, on the radio, totally unannounced. He just started reading this. And people in New York and Chicago all of a sudden heard this, and they thought this was actually happening, and it caused riots in both these cities. And the broadcast was finally interrupted by the military to announce that this was a fictional story and not actually happening at that time. So you can see that there was a lot of interest in the potential that there was life on Mars, and it was very advanced life. Hollywood got into the act. A great movie, Abbott and Costello Go to Mars, in 1953, and I really encourage you to check this movie out at the video store, because it's a totally politically incorrect movie by today's standards, because Abbott and Costello land on Mars and find that it's inhabited only by beautiful young women. In the late 50s, a very noted astronomer observed Mars and reported to see bands of chlorophyll in the reflectance spectra. This is the light that's reflected off the planet. And again, chlorophyll means plants. And this again aroused great interest in the potential that there was life on Mars. And it was eventually shown that this was an artifact of the observational method, and that particular study was withdrawn in 1959. The first spacecraft to visit Mars were the Mariner missions in the 60s, and the photographs that were returned there were startling in the fact that they showed the surface of Mars looked very much like the Moon, a heavily cratered planet, a lot of very detailed terrain, no evidence of water or plants or anything, and this sort of put the gamish on any idea that there was any kind of plant material on Mars. Viking visited the planet in 1976, an incredibly successful space mission. 2 spacecraft landed on the surface, and one of the primary goals of these spacecraft was to search for life. I'll discuss that in a second, what they found. Pathfinder, a recent mission to Mars, landed in 1996. At the same time, it was announced that one of the meteorites that had been found on the Earth was classified as a meteorite that came from Mars, and a lot of excitement was generated about the potential that this meteorite contained evidence of life. And finally, as we enter this new millennium, there's a flotilla of missions that are planned for Mars, and I'll describe some of those in a second. Viking. By today's standards, this is an incredible experiment. The spacecraft itself is a marvel of technology. 2 spacecraft landed at 2 different locations, both with identical instruments. They contained a variety of experiments to test for the presence of biological processes, and they contained an instrument called a gas chromatograph mass spectrometer, GCMS, which was designed to test directly for the presence of organic molecules on the surface of Mars. By today's dollars, this mission would cost in excess of $1 billion. And so, given the constraints of our budgets today, where space missions are in the 300 million range top, we cannot refly a Viking-type mission. We have to be, as Dan Golden said it, cheaper, faster, quicker. If you consider what the Viking results really found, they said that they could not have detected any compounds at the level of 1/millionth of a gram per gram of soil. Now, you might think that that excludes the possibility that there's anything living in the Martian surface, but we now know today that this instrument, even though it was a very powerful instrument, would have missed as much as a million bacterial cells in a gram of Martian soil. So this was a very successful experiment, but by today's standards, we have to do better than this. Our goal is to be able to search for organic compounds at the surface of Mars at the level of 1 bacterial cell per gram, 1,000,000 times better than Viking. The Mars meteorites caused great excitement. How these meteorites get to Mars is very simple. A large asteroid or comet impacts a planet, blows debris off the surface. You can see here rocks being jettisoned from the surface of Mars by an impact event. Most of these fall into the sun, but some of these meteorites fall to Earth, and it's estimated that about 5 Mars meteorites land on the Earth each century. Most of those land in the ocean. A few of them land on the land masses, and one area which has been proven to be very fruitful for collecting meteorites, and especially meteorites from Mars, has been in the Antarctic, where meteorites fall on the ice sheet and are preserved in the ice for long periods of time. To give you an idea how challenging this is the kind of sample you get of these Mars meteorites. Little sugar cube type pieces, and the challenge is to take those little pieces and analyze them for something that might be derived from any kind of organism on Mars. The first reports on this meteorite, which by the way weighed about 2 kilos, was found in 1993. It has a very old age. It comes from the very earliest period of the history of Mars, 4.5 billion years ago. It has these little globules of carbonate in them, which are thought to be perhaps produced by biological processes. It was ejected from Mars 16 million years ago, and it fell on Earth 13,000 years ago. The reports that generated so much interest were that in the meteorite you get these little worm-like features that were claimed to be fossil organisms. Now, the problem here is that the scale here is, this is 500 nanometers. Nanometers that is a billionth of a meter. And these are very small features, and it soon became apparent that to package any kind of genetic material in this size of cell was impossible, at least genetic material as we know it. So immediately the question and criticism arose is that these things are just too small to house the machinery we associate with life as we know it. There were claimed to be some organic compounds in here that were suggested to be derived from living organisms. The problem is these compounds, polycyclic aromatic hydrocarbons, are not part of biology as we know it, and they're readily produced on Earth, and so you had to be very careful that these weren't simply contaminants from our planet. When we approached this problem, we decided to focus on amino acids. That's my area of expertise. And the reason we focused on amino acids is that they're readily synthesized by a variety of natural chemical processes. Many of you have heard of the Miller-Urey experiment, where they sparked a mixture of methane, ammonia, and hydrogen. One of the main products in that was amino acids. And so amino acids are readily produced by natural chemistry. So they could have been present on Mars early in its history. They're abundant in certain kinds of meteorites called carbonaceous chondrites. This was a staggering finding in the early 70s, that these particular meteorites, carbonaceous meteorites, contain a wide variety of amino acids produced again by natural chemistry. More importantly, amino acids are fundamental components of biology as we know it. They form proteins and enzymes. And these play a critical role in our biochemistry. Without proteins and enzymes, organisms could not exist. Most importantly is that you can readily sort out whether amino acids are produced via natural chemistry or a biology, because amino acids have this property called handedness. And just like your hands are mirror images of one another, like if I held my hands like this and I had a mirror here, the image in the mirror would be my right hand. And amino acids could have the same geometry of atoms to make left and right-handed molecules. When you make these by natural chemistry, you make equal amounts of both. Biology on Earth uses only left-handed amino acids. So when we look at living organisms on the Earth, the thing that is very striking is that we see only left-handed amino acids. And the surmise is that life requires one or the other, left or right-handed amino acids. And in my opinion, life can use either. It's a matter of chance. 50% of the time, life in the universe is going to use left-handed like us. 50% of the time, it's going to use right-handed amino acids that we use. So the real interesting thing is if we went to Mars and found right-handed amino acids, that would be a clear indication of unique Martian life. So one of the focuses that we've had is searching for amino acids in Martian samples. And when we did this with the ALH meteorite, we did find amino acids there. However, they were just like the amino acids that we see on Earth, and more importantly, they were just like the amino acids that are found in the Antarctic ice. And so our conclusion is that the amino acids present here are terrestrial in origin and not Martian in origin. And so the bad news with respect to Mars meteorites is they've been greatly compromised by their presence on Earth. So if we summarize this about where we stand right now, early in the history of Mars, liquid water may have been present, and there's even suggestions that episodic transient water may be present even in the modern history of the planet. So water, fundamental component for life, could have been present in the early history, may be present today. Some of the steps that are thought to be important in the origin of life on Earth could have easily taken place on Mars. Viking found nothing at levels that were indicative of a rich biology on the surface, but Viking would have missed 1 million bacterial cells and a gram of soil. Mars meteorites, although they're great to study from an aspect of just bulk geochemistry. Unfortunately, any kind of organic material in these meteorites has been infected by terrestrial material. So they can't really answer this question. And what this all means is that we've got to go back to Mars and do a much better job than Viking did. Now, this is a picture of the 1996 Pathfinder rover. You can see it coming up here and nuzzling up to a rock. This rover is about as big as the top of this podium and stood about this high. So it's about this wide, about that tall. And the challenge here is that putting any kind of instruments on this that could sense for the presence of amino acids that might be associated with life is extremely challenging. The instruments that I use in my lab take up a whole laboratory bench. So you can't fly that on a platform that's that size. So you have to be innovative on how you approach this problem. If you look at what the next decade holds for us, 2001, right now, the Mars Odyssey spacecraft is in orbit around Mars, and if you read the papers, it's just fallen into its final orbit to begin a very detailed reconnaissance of the Martian surface, looking for certain kinds of minerals that might be associated with biological processes. This spacecraft has a camera on it that can resolve things on the surface about the size of a Volkswagen Beetle. Okay, so this is a very powerful camera, and it's hoped that with this camera we'll have even a greater understanding of the surface of the planet. 2003, we'll see 2 rovers land on the surface. Again, this is Pathfinder-sized rovers, small rovers that are designed to explore the immediate vicinity of the landing site. They will not have any kind of life detection devices on them. They're just too small. Another orbiter in 2005. And 2007, this is where the fun begins. A whole series of spacecraft are going to fly to Mars. One are these little airplane gliders that are going to fly through the atmosphere, sampling the atmosphere so we have a better understanding of what the atmosphere of Mars consists of. A balloon that slowly lowers itself up and down, a hot air balloon on Mars to sample various parts of the atmosphere, a device that flies through the upper atmosphere and collects dust. They're going to bring back a few hundred micrograms of dust from Mars. And then you've got this huge rover that's going to land on the surface. This lander and the rover is what we call a monster truck rover. This rover is about 5 to 10 times bigger than the rovers in 2003, and you could literally lay down on the top of this thing. And it's this kind of platform that we're developing in situ instruments. And then finally, the real exciting part here is 2011, we're going to have another similar sized rover. It's going to collect samples, place them in a rocket, and blast them back to Earth. So in the next decade, we will actually return samples from Mars back to Earth for detailed analysis. We can literally throw anything at them that we have available in our repertoire of instruments. Now, how do we go to Mars and do an amino acid analysis that's simple, fast, and small? And when I started thinking about this, I realized that you just had to jettison and forget all of the chemistry that you'd used to analyze amino acids in the laboratory today. And I remembered reading a paper that was published in 1930 about some of the old experiments trying to isolate amino acids from cells where they didn't have all our fancy instruments. And one of the things they found is that amino acids, when heated under a partial vacuum, are volatile. They vaporize, and if you have a cold finger in the system, the amino acids vaporize out of the sample and are trapped on the cold finger. So we made this little simple device, and actually I'm holding that in that picture that Jeff showed on the cover of the Exploration magazine. And what this is a little device. You put the sample in the end of it, you pump this down to Mars ambient pressure, which is about 1/100th the atmosphere of the Earth, so you don't need a vacuum pump. Mars already has a lower pressure than on Earth, so you can just use that. You have a cold finger here, which we fill with liquid nitrogen just because it's convenient. The surface of Mars is minus 100 degrees centigrade, so it's cold. We don't need to cool a device on Mars. It's already cold. And then if you take and flash heat this material, the amino acids that are present in the material here end up being released and captured on the end of this cold finger. And we do the analysis, and you can see here that the amino acids are clearly detected. And our detection here in the lab is we're able to detect a few hundred bacterial cells in a gram of sample using this kind of approach. And so this led to the development of an instrument called MOD. And one of the things I learned in this business is having a clever acronym really goes a long way. And so MOD stands for Mars Organic Detector. And the group of scientists that work with me is called the MOD Squad. And the way this works, and I brought with me an actual prototype model of this so you can come up and look at it afterwards. This is the size of the device. And what this consists of is a rock crusher, which is here, because we're potentially going to get solid samples when we go to Mars, a little oven, which is here, a detector cold finger area, and then another thing on the side of this, which is called a tunable diode laser spectrometer to sense the atmosphere. And what happens is a sample is provided to this arm. The arm then transfers this to the rock crusher, and this rock crusher is nothing more than a series of plates that slowly move together using the power from a AAA battery and slowly crush this sample to a powder. Now, when I've seen this thing work, it sits there and you look at it and goes, God, that thing isn't moving. Well, you have to be patient. You have to wait 6 hours. But it just slowly crushes the thing and crushes it into a powder. The powder drops into this oven area here. This thing is then sealed. And remember, this is all cold. The whole device is cold. And then we heat the oven. The amino acids are released and collect on the cold finger, which is inside here. And then we use fancy laser-based detection methods to sense what's been released. And that's the detector part here. This whole device, you can see, is small, fits in your hand, weighs about a pound. And so this is the kind of device that you could fly on a spacecraft. And so we developed this, and this was selected to fly on a mission that was scheduled to land on Mars in 2003. Unfortunately, that mission has been canceled because of the loss of the Mars Polar Lander. NASA got really paranoid about losing another spacecraft, and so instead of taking a chance and flying a spacecraft that had already been built, they decided to just cancel everything. But the Mars Organic Detector detects amino acids and other compounds directly on the surface of Mars at a sensitivity much greater than Viking. I put down 100 times more sensitive. It's actually closer to 1,000,000 times more sensitive. It also senses what kind of minerals are in the surface using this tunable diode laser spectrometer. Now, since this mission was canceled and rescheduled for 2007, I was asked to take this instrument design the next step further, because MOD, as it's developed and was scheduled to fly on 03, simply looked for the presence or absence of amino acids. It could not tell us what types of amino acids were in a sample or their handedness. And remember, handedness is a key part of assessing whether these compounds could be associated with living organisms. So I was asked to take this to the next dimension of trying to sort out whether these amino acids could be associated with living organisms, and that meant trying to resolve the left and right-handed forms on Mars, given the constraints of the size of the apparatus we had. And we've, again, concentrated on developing a way that can separate these two forms of amino acids and seeing if there's any preference on Mars for one or the other. People have asked me, what would prove beyond a doubt that you had evidence of life on Mars? It would be finding these right-handed amino acids, like I said before. Finding only right-handed amino acids we would be confident is indicative of unique Martian life. Now, what if we found L amino acids, like on Earth? Well, there's two possibilities. Number one, that the second chance that life had chose L amino acids again. Just remember, it's like the flip of a coin. You flip of a coin twice, and you can get heads twice in a row. So maybe this is just the luck of the draw again, L amino acids. Or maybe we're related, which makes it even a more challenging question. So how do you do this in the confines of a spacecraft? Well, again, this is where you have to be really up on cutting-edge chemistry. And it turns out that some unique methods have been developed to do amino acid analyses using some very small micro devices. And here you can see we're separating these left and right-handed forms of a variety of amino acids. And I'm just going to step over here and show you a prototype of the device that we've developed to do this on a mod-type instrument. And this is called capillary electrophoresis and involves, turn that off for a second, involves etching a small capillary channel in a glass plate. Whoops. Oh, the mirror's down. There we go. Okay, so you can see on this plate we have etched a series of channels. And if you look here, you can see this channel here. You can look at this after the lecture. There are a whole series of channels here. We will use one channel on a wafer about the size of a quarter. And with that wafer, we'll be able to apply our amino acid sample that's collected on the coal finger and run it through this channel and actually separate the right and left-handed forms of amino acids directly on the surface of Mars. And again, the size of this device is small enough that it's easily compatible with the kind of size limitations we have for Martian spacecraft. So this is the design of the spacecraft that's scheduled to fly in 2007. One thing I've learned in this business is that you have to be patient because the word is, that I've heard from headquarters, is that this mission will probably be postponed to 2009. So you just have to be patient and wait your chance. And you can see here, this is the spacecraft. Here's the mod instrument sitting in this package right on the side of the spacecraft. This whole platform is about the size of a big table. You notice there's a rocket here. They're going to test with this spacecraft a rocket that will fly off the surface of Mars and rendezvous with an orbiter to test the technology to eventually bring samples back to Earth. So, like I said, this is scheduled here, probably be shifted to 2009 now. And the sample return mission will be shifted probably to 2013. So this is still over a decade away, but eventually we will get samples back from Mars. Now, this generates a problem in itself because there are international treaties signed by all the nations in the UN that prohibit the return of samples from some other place in the solar system without them being quarantined and then certified that they are not hazardous to organisms on the Earth. So collecting these samples is one challenge. Getting them back here is another. The biggest challenge is quarantining them and putting them in a facility where they can be deemed safe until they're studied and proven that they're not hazardous. And this requires an elaborate protocol. Here's the scheme, here's the spacecraft, here's the rocket. Again, acronyms are used throughout this. You notice the rocket's called a MAV. When I heard that, I thought, what the heck is a MAV? Well, that's the Mars Ascent Vehicle. And that goes into orbit to the EEV, that's the Earth Entry Vehicle. The samples are encapsulated in a sphere. This Earth entry vehicle returns to Earth. This is just a direct trajectory right to the planet. This little sphere is released during atmospheric entry, and the plan is for this sphere to land someplace in Utah. Now, you might ask, why Utah? Okay, well, number one, this is a U.S. mission, so we want the thing to land in the United States. It's coming in from the Pacific. The trajectory will have it flying from west to east. The first place that we hope it'll hit will be the deserts of Utah. If it misses that, well, it'll land in Kansas, or it could hopefully land in some place before it would fly off and land in the Atlantic Ocean. Once this thing lands, it'll be retrieved, and this is not supposed to scare anybody in Utah, but these guys in these biohazard force suits will jump out of a helicopter and go and retrieve this thing and whisk it off. And if you think that this is going to generate some sort of panic, I don't know what will. And then this will be housed in a CDC quarantine facility that's more rigidly controlled than the facilities that are used to contain the Ebola virus. Now, there are a lot of caveats here. Number one, this capsule has to be sealed. It has to be sealed on Mars, because the idea is that these samples have to be rigorously contained before they're ever brought back to our planet. So there's going to be a seal that's welded together in orbit. We've never done this kind of stuff, new technology, and so this is all on the drawing boards right now. This thing will be about the size of a big ball, big beach ball or something, and this is what's scheduled to land in 2013 or 2015 in Utah. Once we get it back to the planet, then all kinds of other things have to take place. One of the most distressing things I see is if sample containment cannot be verified en route to Earth, the sample and any spacecraft components that may have been exposed to the sample should either be sterilized in space or not returned to Earth. Well, that last part, not returned to Earth, it's on a straight trajectory back to the planet. The only way it's not going to be returned to Earth is if we blow it up. And I would really hate to see, after going to all this effort, to blow the dang thing up before it gets back here. Integrity of containment should be maintained through re-entry and transfer to the sample receiving facility. That means that we have to hopefully have a technology that will not allow the sample to bust open once it hits the surface of Utah and spill the samples all over the surface of Utah. The samples should be contained and treated as potentially hazardous until proven otherwise. Then this requires a biohazard 4 type facility. If this isn't meant to scare the public, I don't know what is. And finally, samples should be considered hazardous until demonstrated otherwise, and then there should be some controlled distribution of unsterilized samples once they are proven to be non-hazardous. Now, I've been involved in the planning of this, and one of the things I've argued is that you're going to have to release some of these samples immediately to scientists, or you're going to have a revolution on your hands. I mean, scientists want to study these. They want to know what's in them. They want to know what makes up the surface of Mars. And so the efforts have been made to try and figure out how we could sterilize these samples. On Earth, we sterilize samples by either 2 ways, heat or radiation. If you heat a sample like this, you're liable to destroy a lot of valuable information. And so the sterilization method of choice has been radiation sterilization. To give you an example, a lot of our foods are sterilized by radiation. Hamburger is routinely treated with gamma rays to sterilize it from E. coli bacteria contamination. And so the idea is to sterilize the Mars sample with a level of gamma rays 1,000,000 times higher than it takes to sterilize hamburger. Now, this is bizarre in my opinion, because We don't know what's in there. We don't know whether this kind of organism would survive sterilization. It could be something that's used to radiation and not be affected at all. So sterilization is an unknown right now. And this is still being worked out. So you'll read a lot about this. We've got over a decade to plan this. And it's going to be a lot of interesting science associated with just what we do with respect to containment and sterilization of these samples once we get them back to Earth. Finally, I'll just show you this one last picture that's of Europa. This is the moon of Jupiter, where, like I said, there's evidence of a subsurface ocean, and perhaps this is another place we might search for life. There are plans to go to Europa sometime in the next 20 years or so, and again, try and do some of the sensing, remote sensing type analyses we talked about on Mars. And then finally, this is just a picture of our Milky Way galaxy. This is where we are. Look at all these other stars and just think of all the planets that are associated with these stars and the possibility that there's life on these planets. And the big challenge of this next generation of scientists is how do we look at these places for the potential for life? Okay, thank you very much. The question is, we've talked about science. What about the philosophical considerations if we do find life on Mars? I think that's an important issue because, after all, are we unique? Is life unique on Earth? And I think finding life elsewhere would just say that we're a product of natural chemistry, and the potential for life throughout the universe is probably very high. What does it do to religion? Well, I'm not qualified to really discuss that, but I think you can imagine some religions might be quite happy with finding this. Others could be very threatened by it. Can we go to the ice caps on Mars and maybe search for life there rather than in the area of the equatorial regions? In fact, one of the missions that I'm part of the planning team is a thing called a Cryo Scout mission for 2007. And it's a spacecraft that lands on the pole, and it has a device that starts melting its way through the ice cap, and as it melts its way through the ice cap, it is carrying out amino acid analyses similar to what I described here. So that may happen. Well, the question is, how do we sell this to the public with the idea being that maybe the money could be better used elsewhere? The one way I can answer this is that human beings are curious creatures. We want to understand the world. And believe it or not, a lot of technology that spun out of the Apollo missions are directly affecting us today and making our lives better. For example, silicon chips were a direct product of the Apollo missions. And so one of the spin-offs of all of this technology is technology that eventually trickles into our everyday lives and enriches our lives. So I'd say there's two aspects of this. Basic knowledge, understanding our place as human beings in the universe. And number two, the technology spin-off that we'll get from this, I think, makes it sellable. Well, do I have a preferred hypothesis how life began on Earth? Yes. If you read my book, it's the scenario is pretty straightforward. You go from these simple molecules, they undergo polymerization in various kinds of environments. I happen to favor tide pools or frozen solutions where you can concentrate molecules and enrich the chemistry. And as you get more and more complex molecules, some of these molecules, just simply by chance, had the ability to start replicating, copying themselves. And once you get that point, you have the start of life as we know it, because that will evolve through evolution to more and more complex molecules, and eventually the RNA, DNA, protein world that we have today will be a product of that, and then that will explode into all the organisms that we see on the planet today. Who makes up the rules for bringing stuff back to Earth, and can we cheat? This is a UN treaty that was signed by, like I say, all the nations that are part of the United Nations in the 60s, before the Apollo missions. All of the space agencies have what is called a planetary protection officer. And that officer is in charge of ensuring that when we send spacecraft to another planet, that it's sterilized. So we're not contaminating that planet with ourselves. And second of all, when we bring stuff back, that the biology, the ecology of the Earth is protected from potential hostile organisms that we might bring back. I don't think we can cheat on this and really convince the public that this is a safe exercise. Yeah, the question is, what about all the stuff that's fallen on the Earth from Mars already? Hasn't that contaminated the planet with any Martian organism? Well, the answer to that is, number one, I think most organisms would have been sterilized or killed during ejection from the planetary surface and transport through space. Second of all, these things get heated in the atmosphere. Lastly, we don't know whether it's affected us. There are some people that argue that some of our great pandemics of flu and other diseases are organisms from Mars or other places. Now, that's science fiction to me, but we can't exclude that. So this is why we can't say, since we've received this stuff in the past, that it's not hazardous today.